Hi everyone, I am Changting Wei, and today I will share with you this new tactic that could win us the war against resistant bacteria. Yes, when I use the word war, I mean it. Some people compare the recent coronavirus to war. However, bacteria have been killing more people almost since the beginning of mankind. So what can we do to win the war against bacteria? If you look towards the bottom right corner, you see this man called Alexander Fleming. He discovered antibiotics, which became our main weapon against bacteria. These brave soldiers can kill bacteria rapidly and not cause harm to our bodies. However, we quickly discover that antibiotics are not working so well. These are due to the mutated bacteria that have developed a resistance against antibiotics. Mutations are changes in the DNA that happen randomly. Just like those mutants you see in the movies like X-Men, these resistant bacteria are like super soldiers that can no longer be killed. We have to then upgrade our antibiotics and try to kill them again. This process happens again and again. What doesn't kill them makes them stronger, and each time we try to upgrade our antibiotics, the bacteria just gets stronger. Millions of people die per year solely due to antibiotic-resistant bacteria. By 2050, it is estimated that 10 million will die every year. Just to develop stronger antibiotics will require a fortune. Antibiotics might have served us well for decades, but with them, we are just fighting a losing war. So is this the end? Are we bound to lose? Scientists at KU Leuven in Belgium, led by Professor Hans Steenackers, aren't ready to give up the fight yet. They ask the question, if antibiotics aren't working, why not we change our tactic in fighting these bacteria? Divide and Impera, or better known as divide and conquer. Great war leaders such as Julius Caesar and Napoleon have used this war tactic to defeat large armies. They take the armies, split them into smaller groups for attacking, and eventually take them all down. Now you must be wondering, how on earth can we use this war tactic against humans and apply them to the war on bacteria? Well, let's first understand how bacteria work and why they are so strong. Firstly, bacteria form armies. Strength comes in numbers, and somehow bacteria have figured that out. How, you might ask? This comes to the second characteristic. Bacteria can form something like an army camp. This is where they share public goods, which is the actual term used when bacteria can share certain resources with one another. For example, some bacteria can produce goods that are like shelters in army camps. By sharing them, they can protect each other and become stronger as a group. One of these essential supply of public goods is EPS, or biofilm. You know how there is this super hard plaque on your teeth that's just so hard to remove? That is one example of how it works. Some bacteria can produce this exopolymeric substance, or EPS, which is a slime-like substance, or biofilm, and share it with the rest. This holds them together, much like the army camp I mentioned before. These can also act like a barrier, protecting them from external forces, just like how we can seem to remove the plaque just by brushing our teeth. EPS is essentially what binds them together and makes them stronger. The scientists decided to use Salmonella in their experiments, as it also produces EPS. It is one big factor of food poisoning that affects many of us when we eat uncooked food. EPS and Salmonella have three functions. 1. It can form plaques like those on your teeth. These are formed on petri dishes in the lab. When they are exposed to disinfectants and chlorine, only those with EPS production can survive. 2. They require extra resources as they cannot form these when there is not enough food. 3. They can share EPS with each other. When scientists put salmonella that has EPS with those that don't, they all group together and shared it. So if EPS is the problem, why don't we stop it? To do this, we must go to the root cause of how EPS is formed. The scientists went straight to the origins, the genes. Gene CSGD is the one responsible for producing EPS. Much like a factory, the inner workings of production of EPS has to be a certain way. The scientists have found a chemical substance, which we will just call EPS inhibitor. This essentially stops gene CSG and stops all production eventually. Without this line of production, EPS are no more. So the scientists tried to add EPS inhibitor to salmonella and see what happens. The result? No more EPS were formed at all. The bacteria were left alone to fend for themselves. If we use disinfectants or chlorine now, we could easily kill them off. However, in order to further understand how this works, the scientists did not stop there. When they decreased the dosage of the EPS inhibitor, there were some, but few, that produced EPS. Just like those antibiotic-resistant bacteria. By chance, there are some that mutate and become super soldiers, which means they are no longer affected by the EPS inhibitor. To further experiment on this, the scientists took these mutant-resistant soldiers and mixed them with non-resistant ones. After a while, they discovered that all the mutant ones started to die out. They realized that sharing of the EPS is too costly and such waste of effort to share amongst those that didn't actually need them. This also shows that even if some do become resistant to EPS inhibitors, it is not like antibiotic resistance. They actually become weaker because they are trying to bring together a now divided army all by themselves. This gives us sufficient time to kill them off all together. So what have we learned? Bacteria uses EPS to bind together like army camps, which makes them stronger. EPS inhibitor that the scientists found divide them up. Then they become easy to kill. Even if some survive and can produce EPS, they are still weak on their own. Doesn't this sound familiar? This is divide et impera in play, and this new tactic has now been proven effective. So what's next? To quote Professor Steinakers, the next step is to introduce this into clinical practices, such as pills or coating on implants to prevent infection. Though this might take some time, we now have hope to turn our tides in this war on bacteria. For too long, they have been affecting our lives. For too long, they have gotten past our best efforts of attack. For too long, we have been fighting a losing war. No more, we shall say, for victory is ours, and this EPS inhibitors will be our way forward. Thank you.